At this ASH, we presented results from the Universal trial. This is a phase 1 trial of ALLO-715, which is an off-the-shelf allogeneic BCMA CAR T cell in combination with ALLO-647, which is a anti-CD52 monoclonal antibody for patients with relapsed and refractory multiple myeloma. So what's new about this? This is the very first off-the-shelf CAR T cell, off-the-shelf being this is a donor-derived CAR T cell and has several potential advantages. One is there's no need for customized manufacturing, therefore no wait time for getting on the study. And this is a huge advantage for patients with relapsed and refractory multiple myeloma. In fact, we show in the study that about 90% of patients on the study were able to get their CAR T cells or were able to start protocol treatments within 5 days of enrollment to the study, which is a huge change from about 4 to 6 weeks of wait times for autologous CAR T cells. Allogeneic CAR T cells also mean we can do bulk manufacturing. The product variability is lower. There's a possibility for repeat dosing. So it has several potential advantages. So the field has been interested in knowing what the experience with these off-the-shelf CAR T cells would be. And this is the first report. Here we report results from the first 31 patients in a dose escalation fashion. Patients were treated in doses of 40, 160, 320, and 480 million CAR T cells over the last 1 year and a couple of months at this point. What we're showing is that it's feasible to do off-the-shelf CAR T cells. As I mentioned, we are able to deliver these treatments fairly quickly after enrollment in the study. We also show that none of the patients required bridging therapy prior to getting the ALLO-715, which again is a big advantage in this patient population. And coming to safety, I think we show that this is safe to administer. So we saw the expected toxicity with this treatment, that is CAR T cells, the cytokine release syndrome, which was seen in about 45% of our patients, but they were all grade 1 or 2 and managed with minimal interventions. Only less than 20% of patients required tocilizumab or steroids on the study. Notably, we did not see any neurologic events or graft versus host disease, which would be a concern with allogeneic CAR T cells. So far with 31 patients treated, we did not see either of these toxicities. We did see some infections, including grade 3 or higher infection. One of the patients in the study died from a grade 5 presumed fungal pneumonia, which we attributed to his progressive disease and lymphodepleting chemotherapy, including ALLO-647, but not to the CAR T cell itself. And lastly, I guess efficacy. We do see good early evidence of efficacy with this product. So at the higher dose of 320 million cells, there were 10 patients treated with the standard lymphodepletion at this dose level, and 6 out of the 10 patients had a response. And some of these patients are now having ongoing responses at 6 and 7 months. And several of the patients at these higher doses that had a response were also MRD negative by flow cytometry, so again showing an early sign of efficacy for this off-the-shelf product. Where do we go from here? I think the follow-up is still very short. The median follow-up at the time of presentation was 3.2 months, particularly for the higher doses. We need to follow patients for longer. The study is continuing enrollment. We're enrolling patients at the higher doses of 320 and 480 million cells. We're planning to test different doses of lymphodepletion in combination with these higher doses of cells. So those are patients are being treated currently and in the next few months. And then really, I think, waiting to see what the long-term follow-up looks like for these patients who are treated at the higher doses.